Hello, fellow faculty, students, and judges. My name is Charlene Mansour. I'm an undergraduate student at UAB, majoring in genetics and genomic sciences, as well as cancer biology. I work in the lab of Dr. Ojesina within the Department of Epidemiology at the School of Public Health. And so today I'd like to share my research with you that is focused on investigating the effect of bacteria in the growth of breast cancer cell lines. So as you may know, breast cancer causes a whopping 15% of deaths in women worldwide. Just as there are bacteria all over our bodies, recent studies have shown that there are a diversity of microbes in breast tissue that play important roles in both health and disease. However, the role that bacteria plays in influencing breast cancer is not very well understood. And so in this project, our aim was to basically understand the role the microbiome plays in breast cancer, specifically in relation to HER2-positive breast cancer and its response to trastuzumab treatment. And so as you can see in this chart, depicted are the relative frequencies of various types of breast cancer that were studied within TCGA. TCGA is the Cancer Genome Atlas. It's a joint effort between the National Cancer Institute and the National Human Genome Research Institute. TCGA comprises a vast population study of 11,000 patients across 33 cancer types, one of these types being breast cancer. We focus specifically on HER2-positive breast cancer, which is important because this is a subset of breast cancer for which treatment has been developed. And so this treatment is known as trastuzumab and goes by the common name of Herceptin. However, in spite of this, of HER2-positive patients, only 50% respond to trastuzumab treatment. And so we hypothesize that differences in bacterial content of patients may influence and determine those who respond to treatment versus those who don't. Hence, our overarching questions for this experiment and this following project are there bacteria that encode or resemble human-like HER2 sequences? And 2, do HER2-like kinases make it less likely for tumors to respond to treatment? And so one of the ways to identify bacteria in human tissues is to perform sequencing and to computationally subtract out human sequences to yield residual pathogen sequences. This is analogous to separating metallic paper clips from plastic paper clips using a magnet. And so we applied this method to TCGA. Specifically, we applied this sequencing method to HER2-positive breast cancer and its adjacent normal tissue. And so as you can see in the figure, acidobacteria is more abundant in HER2-positive breast tumors versus other bacteria such as Ralstonia that are more abundant in normal breast tissue. And so this is a comparison of bacterial abundance from the same individuals. Based on these results, which indicated again that acidobacteria are more abundant in HER2-positive breast cancer tumors, we performed a blast alignment. This blast alignment allowed us to compare the human HER2 protein kinase and the acidobacteria protein kinase. The acidobacteria protein kinase will be further abbreviated as APK. And so what we did next is we designed a construct to express APK and HER2 in breast cancer cell lines. We monitored the growth of cells over 4 days. And after 4 days, the cells with the empty vector grew 3-fold compared to the first day, and the cells with HER2 grew 3.5-fold, which is a higher rate than the empty vector and was expected. But what's fascinating is the cells that were treated with APK grew 5-fold. And so this suggests that APK plays a role in increasing the growth and proliferation of breast cancer cells. So in summation, we discovered a bacterium that encodes a HER2-like kinase and is overexpressed in HER2-positive breast tumors versus normal breast cells. The HER2-like kinase, which we tentatively called APK, increases the proliferation of breast cancer cells. And so these results are actually very encouraging. They have motivated us to pursue future investigations that repeat these experiments in different cell lines. And so if you look at the table, specifically where the red check marks are placed, our results thus far are from experiments in HER2-negative cell lines that have not undergone treatment. And so what we wish to do is to conduct further experiments in HER2-positive cell lines that test the effect of this treatment. And so when treated with trastuzumab, we expect that HER2-positive cells should reduce in proliferation. This is because trastuzumab is a monoclonal antibody that targets HER2 receptors. And so we hypothesized that if APK plays a role similar to HER2, then the APK cells should also reduce in proliferation in the same way that HER2 cells do. Ultimately, if we're able to prove that APK increases the proliferation of HER2 positive breast cancer cells, this work will have established a foundational proof of principle that bacteria play a role in cancer growth. In view of the public health importance of breast cancer, these results will also have clinical applications. They may account for some of the patients who do not respond to treatment, as I previously mentioned. And hence, what we can do is we can spearhead the efforts to develop novel treatments that target bacteria that play a role in the development of breast cancer, and ultimately improve the prognosis of these patients. In closing, these studies were made possible through support from the V Foundation as well as the National Cancer Institute. As well, I'd like to thank the O'Neill Comprehensive Cancer Center and the School of Public Health for providing us with the opportunity to conduct this study. Thank you for listening to my presentation. I hope you enjoyed it and feel free to ask any questions.
Hey, Charlene, that was really well done. So I do have a few questions to ask.
Absolutely.
First, I wanted to know, you said you, when you were looking into the bacteria, sorry, my headphones just fell down. When you were looking into the bacteria, you found, let me just get it right, a Acidobacteria and Acidobacteria, right?
Just acidobacteria, just one that we studied.
You only studied that one. So why that one in particular? Like when you separated it, was that the only bacteria you found?
Yeah, so I can actually share my screen again if that's okay. Okay, so I think it went away. There we go. All right, so what we initially did was, so this is a kind of complicated figure, but essentially all human disease tissue has human cells and it has non-human cells. And so what we specifically tried to identify was bacteria, because we had prior analyses, we found that bacteria could cause the increase in breast cancer cell proliferation. And so what we did is we basically had diseased human tissue, and we subtracted all the things that are human from it. And so, like, as you can see through this little magnet, we took out the human genome. And what we were left with was a bunch of pathogen sequences. And so from these pathogen sequences, we analyzed them and sequenced them, and we found a specific bacteria, which was the acidobacteria, that was more prevalent in HER2 positive breast cancer cells. And so with the Ralstonia, that one is just a representative of bacteria that is normally present in breast cancer tissue. In normal breast tissue, and acidobacteria was present in breast cancer tissue. So we found that, hey, like if this bacteria is present more in HER2 positive breast cancer tissue, perhaps it's causing the proliferation of these tissue. And so what we did further is we took HER2 and we took the acidobacteria and we compared both their amino acid identities. And so this picture is a little complicated, but I'm not sure if you can see my mouse, but essentially you have the query, which is the HER2, and the subject, which is the acidobacteria. And so we compared both of these. And so in between both these query and subject lines, you'll find random letters. And so these letters essentially mean that if it goes LLL, that means leucine is present in both the HER2 and the APK, and therefore they have at this position the same amino acid. And so as you can see, they have quite a few similarities. And so that's why we chose to kind of look at HER2 and APK and compare them. I hope that answers your question.
It does, thank you. And my next question is, so how, you said that the control was basically a breast without cancer, right? The same person, a breast without cancer.
Yes, sorry, continue with your question.
Oh, sorry. If that's the case, how early on do you start seeing higher concentrations of acidobacteria?
Yeah, that's a great question. So Um, we had a couple controls. So one of the controls for the specific experiment where we developed and found acidobacteria was looking at normal breast tissue. Other controls were that we actually developed these experiments in HER2 negative cell lines that were not overexpressed. Um, and so we didn't specifically work with patient populations, we kind of just worked with their cells. And so I'm not sure how early on they developed, but we got kind of the final product of the development and we analyzed that, if that makes sense.
Okay. And I don't know if you'll know this if you've only worked with the cells, but um, if uh, uh, if you were looking at the cells during chemotherapy, um, what kind of effect did chemotherapy have on the pathogens? Like, did it kill all of the pathogens? Did it only kill the acidobacteria? What happened?
Yeah, that's a fantastic question. So actually, as I mentioned, this work is actually pretty novel. No one really knows how bacteria impacts breast cancer growth. And so with that, we also don't know how treatment impacts bacteria. But what we do know is that since chemotherapies affect cancer through certain pathways, such as, for example, through certain receptors or so on and so forth. If these bacteria are increasing proliferation of cells, then most likely chemotherapy will not kill them off because it doesn't target that specific pathway. And so that's really interesting because if bacteria are contributing to this growth of cells, then we'll have to develop novel treatments to target both the cells and the specific chemotherapy pathways as well as the bacteria.
So, do you think, um, just from curiosity, and if you know, what is the chance that after somebody's been treated, like after somebody's gone into remission from breast cancer, what is the chance that they'll um, have breast cancer again?
Yeah, that's a good question. I'm not sure of the actual percentage. It all is variable based on one, when the cancer was identified. Was it stage 0 to 4? The later it's identified, the more likely you'll have very tough treatment and like it will be targeted very hard. And with that also comes metastasis. So if it has metastasized to other parts of the body, then you will have likely a more probable chance of developing the cancer again. So again, it depends on the stage. And what is used to treat it. Is it radiation? Is it chemotherapy? Because we know chemotherapy is almost like a systemic drug, which targets almost like every cell in your body. And so if it's chemotherapy, then that might be different from radiation, which is just targeting one specific site, and there might be a metastasis in the body. So it really depends on the variables and the patients we're looking at. But depending on those different things, breast cancer is not one of the highest known to have reoccurrence. There are more cancers that are more likely to reoccur. And so I hope that puts you a little bit of peace in mind, but there is always a chance with cancer that it will come back.
Okay. Well, those were all my questions. Thank you. That
Thank
gave
you
me a
so
lot of
much.
clarity. And I'm
I'm
also
glad I
really
can help.
excited to see what you guys do with this in the future.
Thank you so much. Yeah, I'm really excited. I think it's like pretty novel work. And so I'm super passionate about breast cancer. So I can't wait to see where we go with this.
Good luck.
Thank you.
Does anybody else have any more questions?
No, I was just gonna say great job. Very interesting.
Thank you so much.
Mm-hmm. Okay, well, I think you're free to go. Right, Nilu?
Yeah.
Okay.
Unless you have
Awesome.
anything else to say, you're good.
Thank you all so much for listening. I really appreciate it.